Our next presenter is Dr. Jose Abisambra. He goes by the name Joe, and he's from my state of Florida at the University of Florida in Gainesville. And the title of his project is Tau ribosome interactions are a fundamental pathogenic event. And Dr. Abisambra is the recipient of a $200,000 CART grant this year.
I want to, in true fashion, thank you not only for believing in the project and your support, but also because I just want to share that this award means a lot to me because, as I mentioned earlier, before we started, I was affiliated with the Rotary Club when I was in Colombia, living in Colombia, which is my home country. And I had very formative experiences thanks to the Rotary Club. So this award means a lot to me scientifically and personally. So from the bottom of my heart, thank you very much. And from there, I'm glad you read the title, which is a mouthful. So I'm going to just, for my own simplicity and love of my country, going to change the title a little bit. And although it's a little dramatic, of tau ribosomes and other demons, it pays respect to my compatriot and former Nobel laureate Gabriel Garcia Marquez, who wrote of love and other demons. And he unfortunately, at the end of his life, suffered from cognitive impairment as well, believed to be Alzheimer's disease, but not quite confirmed. The main objective of the proposal that you are supporting is at the essence of the work that we do, and it's to establish how Alzheimer's disease starts. And this is one of the major questions that plagues the field. One of the major challenges is that we really don't know how it starts. And if we can identify this, maybe we can find very effective strategies to intervene and help the lives of more than many individuals with Alzheimer's disease, and as I'll explain later, folks suffering from tauopathies, which is a larger umbrella of diseases that are associated with Alzheimer's, and these amass more than 30 million people worldwide. Not only identifying them, but we're going to leverage this project to further develop some technology in adapting for early diagnostics with MRIs. And I'll show you a little bit about that at the end of the talk. And we're going to follow up on a previously tested strategy in our lab that helps overcome some of the challenges, but and hopefully propel it forward much more effectively. So I think we know a lot about Alzheimer's disease in terms of what we've learned over the past 100 years of its pathology. And whenever we think of a patient with Alzheimer's, I think we've all recognized that we probably know someone or knew someone who had Alzheimer's and they have cognitive impairment, dementia, and an inability to form new memories, memory decline, cognitive decline, and learning. This is one manifestation of the disease in an artist, William Utermohlen, who you can see in 1967, this beautiful self-portrait. He was diagnosed with Alzheimer's in 1995, and he vowed thereafter to do a self-portrait every year. And you can see how Alzheimer's disease has devastated the brain of William. And you can see not only the manifestation of a loss in perhaps perception, but also use of color is something that struck me. Depression seems to align with Alzheimer's symptoms, and maybe motor deficits, which also seems to be in some cases a problem in patients in later stages. So Alzheimer's disease was originally described over 100 years ago by Dr. Alois Alzheimer, and he was a psychiatrist and neuropathologist. And when his first patient with the disease, August Dieter, passed away, he was able to take the brain and stain it. And he identified two major pathological signs of the disease. These extracellular toxic plaques that build up, they're composed of this peptide A beta, and the intracellular aggregate of this protein called tau, which has a normal function, very important, but in disease it adopts this abnormal, toxic, nasty conformation that ultimately builds inside these neurons. And as I say colloquially, it just chokes them. And it's so sticky and hard to break apart that here, this is a staining of tau and green in an Alzheimer's brain. And you can see these flame-like structures. In many cases, these are called tombstones or ghost tangles because these neurons are already dead. And all that survives is this tau aggregate. Now, I'm very interested in tauopathies because they're not only, Alzheimer's disease is the major tauopathy, but this tau pathology exists in the brains of people with more than 25 neurodegenerative disorders. And not only do they share this pathological hallmark, this aggregation of tau inside neurons, but they also share the fact that virtually every patient suffers from cognitive decline, problems in learning and memory. And so let's go back and we started thinking about this. Well, what is a memory and why does it fade? Why does it get broken? And in many ways, the easiest way that I think about memory is in our everyday, our computers. They have storage. We can store past experiences in our brains, and we have an ability then to access these memories. But our brain certainly is much more complex than the power to access the memories, to store them, to form them. So I'm going to refer to Dr. Eric Kandel, Nobel laureate, to better explain how important it is to have a strength of communication. With his colleagues, Arvid Carlson and Paul Greengard, they won the Nobel Prize in studying learning and memory. And I want to highlight that their point of strengthening communication between nerve cells, between neurons, is really a highlight of healthy learning and memory. So when we think about memory, it's beyond just the macro understanding of the brain. It's how it's broken down to the cellular level. And Sue did a great job at highlighting the different kinds of cells. Here, I'm going to focus simply on neurons and highlight that the connections in many places form at synapses. So these synapses can be strengthened simply in the way that we've been thinking about this problem by the formation of new proteins. And so new protein synthesis in a coordinated and exquisitely regulated fashion can form different types of chemicals, such as neurotransmitters, receptors to accept these transmitters, to communicate these chemicals, to communicate between cells, scaffolding proteins to hold the membranes together and the receptors right at the membranes, and finally enzymes that can regulate if there's too much or too little signaling. They can help get rid of some of these chemicals. But importantly, these new proteins have to make ribosomes. And this is where a lot of our work focuses on, is ribosomes, because they are, and part of my drama and romanticism, the birthplace of memories, I think. And it really is, because here's where new proteins are made. In a process of translation, RNA comes to the ribosome, and during this process, we get new proteins. And again, we get the formation in a very carefully regulated fashion of all the types of proteins that I mentioned, all the chemicals that allow neurons to communicate at synapses, and the structures that are important to regulate them. Now, in 1987, Dr. Skip Binder in his lab identified that toxic tau in this hyperphosphorylated, nasty, sticky form associated with ribosomes. And so RNA could come to the ribosomes, but the consequences of the tau ribosome interaction were unknown until 30 years later. And corroborating these results, we've started to study this problem and answer this question: what happens? And we found that, at least using in vitro assays, that there's a decrease in translation that led to an imbalance. So here, for example, we see a decrease in the chemicals that are made, decrease in the enzymes, decrease in ribosomes, actually, receptors. But then there was overrepresentation of other proteins, such as some of these scaffolding proteins. And finally, an increase in stress proteins, such as HSP90, just as an example, highlighting how the cell, the unit, is trying to compensate and survive this assault on the cell that may last many, many years, till a point where it can no longer compensate for it. And so we find ultimately decreased protein synthesis, weakened synaptic strength, and impaired learning and memory. So part of what we're trying to do is first in this project is to establish which proteins are synthesized, not only which ones are decreased, but also which ones are increased, and try to rebalance that. Can we rebalance the change in the amounts of proteins? And then second, and certainly that's the second part of the grant. So the way we're going to do this is thanks to new technology that has maybe not so new anymore, but certainly the types of approaches we can use are really exciting. Now we can answer these questions. So one of the experiments we're going to do first is taking advantage of the brain banks that we have access to. And we've worked with folks here at University of Florida, and also in my previous institution at the University of Kentucky. Dr. Peter Nelson there is going to provide a lot of brains for us. And so will our new neuropathologist here at UF, Dr. Stefan Prokop. And we're going to obtain brains from folks who had Alzheimer's and healthy controls, and isolate ribosome RNA complexes. And once we do that, we're going to cut RNA that's hanging around, and the RNA that's preserved will be protected by these ribosomes. And we know that these ribosomes are manufacturing proteins that are coded by this RNA. So now we can find out what is actually made in these brains. We can get rid of the ribosomes. Now we can isolate the RNA and do some very specific RNA-seq. We can sequence the RNA, which will identify which ones are the proteins that this ribosome was trying to make. And we are going to get two pools. We've done this in mice. That's mitigated a lot of the risk of this project. And we can identify differences in the types of proteins that are made in healthy brains and those in Alzheimer's disease. And we'll find some that are increased, and importantly, we'll find many, many others that are decreased if they follow what we've done already in our animal models of Alzheimer's disease, of tauopathy. And so while we identify them, we can now also explain the processes that lead to this weakening of synapses and connections, and how that may affect memory and learning. And ultimately, in the second part of the project is we're going to try to rebalance translation. And doing that is by identifying the pools of RNAs that are decreased and then supplement them in the brains of mice. And this will be done using AAVs and other technologies that we have available to us. Once we give these RNAs, what we're going to do is run the mice through different outcome measures of learning and memory. We have a multiphoton microscope to measure function in vivo in a mouse that's alive. We can actually see it through a window, a cranial window. We're also working, as I mentioned, on a new adaptation of MRI to measure function, calcium-based function, and finally do some memory testing. And just to show you an example of a strategy that we recently tested to rescue translation, using this compound in a partnership we had with GlaxoSmithKline, working with this compound that rescues translation. It helps improve translation. And here we have mice that are a model of tauopathy. These are normal mice. And I want to highlight this structure right here. This is the hippocampus. And this is one of the most important places where we get the original or the initial parts of memory that is essential for that memory to begin to form. This is a transgenic sick mouse. Look at how thin that hippocampus is compared to these two hippocampi, very plump in the non-transgenic healthy control mice. This hole here means that there's such devastation and neuronal loss that now we can see actual holes. This is CSF filling in those holes. But look at what happens when the transgenic mouse is treated with this drug that rescues translation. You get almost a complete recovery, and statistically, it's indistinguishable from the controls. So we're very excited about this. The next test that we're going to run, or series of tests, will be behavioral. Here, I wanted to show you this cool test. It's kind of a measure of anxiety, and it's based on an open field test. So basically, we'll put the mouse in the middle of a box, and these red lines represent where the mouse is running. And if the mouse is running on the outside, it's thought, or it's interpreted as this mouse is really anxious, protecting itself from a predator coming from behind. But when we treat the transgenic mice, the sick mice with the drug, this mouse doesn't seem to care much where it's running. It seems to be more in the center and this corner. Doesn't seem to be much of a problem. So just to wrap it up, our project again is focused on identifying how Alzheimer's disease starts. We're hoping to identify these missing mRNAs, supplement them to rebalance, and identify how the disease starts. What are those processes? And better explain. And here we can do something really exciting to me personally, is also to understand how the normal process of memory occurs, and certainly how it breaks. We're going to be able to hopefully leverage MRI technology using these manganese contrast agents, which are fairly primitive, but we're hoping to use this project to further develop them and follow up on this strategy of increasing translation. None of this could have been done without the lab certainly building up to this. Much of this work is inspired by my former postdoc advisor, Chad Dickey, who passed away. Pete Nelson gave us the brains, Jada Lewis, who gave us the mice. Also, Peter Davies, who recently passed away, gave us a lot of the tools and really believed in our work, shared a lot of his antibodies. Rakesh Kayed, some folks at the University of Rome have helped us develop some of this. Our funding from NIH. And I'm really excited to put our most recent support from you in some of the work that we're going to develop in the future. And we talked a little bit about gators earlier. These two fellows here are outside of my office in a little pond, and I get to see them every day, and it doesn't get old. So every day I take a picture. I'm not sure what happened to this one. This one seems to have disappeared, but it's a striking feature of the University of Florida.
Jose, thank you so much. That was a fabulous presentation. And there you go. So Dr. Abisambra, Bill Parker and I are just thrilled to death to present you a check for $200,000 to support the work you just described. So congratulations, and thank you so much for submitting your proposal.
Thank you so much for your support. It means a lot.